Welcome everyone to today's webinar. My name is Marie Latulippe and I'm the Director of Science Programs at the Institute for the Advancement of Food and Nutrition Sciences, or IFANS. And hopefully many of you have joined our previous programs, but for those of you that might be less familiar with IFANS, we are a nonprofit organization focused on advancing food and nutrition science for public benefit. And the way we do that is we connect our tripartite membership, so that's scientists from government, industry, and academia, to support collaboration and to catalyze knowledge in those areas. And we also, as IFANS, fund actionable research. That research is conducted by external individuals who are selected through an open and competitive process. And if you're interested in learning more about all the projects that IFANS is currently funding, those are listed on our website. We also conduct our work by leveraging pooled funding that is combined with in-kind contributions from both the public and the private sectors. When you registered for the webinar, hopefully you noticed that IFANS is an approved provider of continuing education by the Commission on Dietetic Registration. So after the webinar, we'll provide everyone with access to the certificate. And just a brief note on logistics before we get started. Hopefully you see a control panel like this one on your desktop, and it should have a questions box that you can expand. What we'll do is we'll go through all three presentations, and then we'll open this up for a dialogue at the end. But as questions come to mind throughout the program, please go ahead and type those in, and we'll start to organize them and get ready to answer. All right, so let's start by introducing our moderator for today, and that's Dr. Johanna Dwyer. If Johanna, you'd like to join us on webcam. And Dr. Dwyer is a professor of medicine and community health at the Tufts University Medical School. She's also a professor of nutrition at Tufts Friedman. In addition to that, she's a senior scientist at the Jean Maillet USDA Human Nutrition Research Center, which is at Tufts University. And Dr. Dwyer has served on many committees, including the 2000 Dietary Guidelines Advisory Committee, and she was also elected to the National Academy of Sciences in 1998, among many other honors that we don't have enough time to cover here. So, Dr. Dwyer, we are pleased to turn the platform over to you.
Thanks, Marie. It's great. Can you put up my slide with disclosures on it? Thank you. These are my disclosures. And let's just get to the program. We've got three wonderful speakers today. And what we'd like to do is, first of all, provide you with an understanding of the state of the evidence on the value of live dietary microbes for health, with the idea that there may be value in ultimately developing a recommendation level for intakes down the line as the evidence evolves. Today's speakers are going to review what we know today about the value of dietary live microbes, and then what kind of evidence would be needed to establish a recommended intake. And finally, what we can do practically right now to leverage the existing knowledge. So let's get right into it and start off with Dr. Joanne Slavin, who's a dear friend and a wonderful scientist. She's a professor in the College of Food, Agricultural, and Life Nutrition, Natural Resource Science at the University of Minnesota, Twin Cities. And she's also published more than 350 scientific articles with particular expertise on the gut microbiota, dietary guidance, in the role of diet and disease prevention, and of course in dietary fiber as well. Joanne was a member of the 2010 Dietary Guidelines Advisory Committee, and she grew up on a farm in Wisconsin, which she still owns and manages. I think it's a dairy farm. So Dr. Slavin's going to be covering the rationale for the hypotheses that live microbes in the diet are relevant to health benefits, and then what evidence you'd need to advance the dietary guidance. Joanne?
Thank you, Johanna. I will first give you some disclosure. It's really nice to be here today sharing this information. These are some of the research grants that have funded my research, and I really appreciate in the fiber world and this idea of real foods, people that are willing to fund research. I'm on the scientific advisory board of different groups here. You'll see it's quite carbohydrate focused, but also a little bit on protein and fat. And I work at the University of Minnesota, University of Minnesota Twin Cities, and I also own our family farm, which I grew up in down in Walworth, Wisconsin. So here's just a little bit about, you've already heard from Johanna, my background, but really appreciate getting involved more, coming from fiber and trying to get together with people and come up with recommendations for live microbes. I always like to start with this slide just so people understand it's a long ways to get to recommendations, and it doesn't happen overnight. So on the left on the screen here, we have the dietary reference intakes, and I teach advanced human nutrition, and I always tell my students those are the recommendations. It's kind of like the nutrition Bible, basically settled science. We have enough agreement that these vitamins are essential. We know what the RDA for protein is. We know what the RDA for minerals are, and as we get into different components, we have to use a similar process to try to figure out, are they required nutrients or food components? How do we figure out how much we should recommend, and what are the health attributes of those components? In the middle is the dietary guidelines, and we're really fortunate in the US to have the dietary guidelines every 5 years. A panel of experts gets together and comes up with dietary guidelines for Americans. So I was on the 2010 Dietary Guidelines Committee, and that orange book there is the scientific background for the diet and disease relationships that were evaluated in the Dietary Guidelines for Americans. Very open process right now. We're going for the next iteration of the Dietary Guidelines, and you can submit information, and other countries really have just in awe that we have that ability to do that. And then we get to the plate, which is how do we get those nutrients into food and give people recommendations. And each of these are important steps in that process of taking the science we know, what are the requirements, and putting it into practice for us. So what are the DRIs? Those are those blue books over on the left. They go back, they're like 20 years old, and they can get updated if there's new information, but they don't have to get updated if there's no reason to change those. So it's a set of reference values for the intake of energy, nutrients, and food components that we can use for planning and assessing the diets of healthy people in the US and Canada. And I want to point that out. This is a shared resource between the US and Canada. We do address both excess consumption as well as nutrient deficiencies, and it has a very broad reach. Promote good health and reduce incidence of chronic diseases. So I use this from my book, Recommendations for Nutrient Intake. So this dietary reference intakes is a huge umbrella term for all of these recommendations we have for nutrients within the US and Canadian diet. And over on the left, you'll see different categories, and this makes it kind of confusing, but I want to go over to the energy intake recommendations because I appreciate it's a whole nother, how many calories do we need? And obviously these are going to be the average amount of calories. We're not going to overestimate there. We want to go, obesity is a huge problem. So in that whole recommendations, we want to come up with the estimated energy requirements for an average person across the life cycle. We also have these acceptable macronutrient distribution ranges that are over in that category. And that seems to get confusing to people, but generally carbohydrates are most of our diet. Proteins 10 to 35%, depending on calories. Fat 20 to 35%. So those are there. On the left, nutrient recommendations. And first of all, we need to figure out, do we have an average requirement for a nutrient? And then we'll show you how to scale up to get an RDA. But we have this other category, and as somebody that's worked in dietary fiber, adequate intake is a place where we cannot come up with an average requirement. So we do basically a scale up or an estimate of what is the requirement with a different process. And then we also have these ULs, tolerable upper intake levels, because we know not all nutrients are 100% safe. They can actually cause problems. They're not just good, they may cause problems. So here's just how we get an RDA. And when people say, hey, let's get an RDA for live microbes, thinking that's a walk in the park and pretty easy, it's a very heavy lift to get an RDA. So first of all, we have to have an estimated requirement. What's the estimated requirement? And you can see that on the graph there. If we can get that, knowing what does the average person need for this nutrient. And typically a lot of the nutrients that have RDAs to prevent a deficiency disease. So we have data that shows this is the average amount that most people would need to get to make sure they don't get the deficiency disease that's related with that. When we set an RDA, we set a big factor. We scale up to, so we go over, so 98% of the population should meet that. So it's an allowance, it's not a requirement. It's very confusing. So obviously, with an RDA, most people in the population should be covered. And then we also have this UL, which is set way wherever it needs to be. And that is, all of this data is bringing together experts and coming up with these numbers. And at a UL level, are you going to have a problem? Potentially not, but potentially yes. So a lot of nutrients are set for, there is a UL because there's a potential negative. So I like to start with fiber just because that's when people came to me for live microbes and they said, hey, how did fiber get an RDA, or does it have a requirement? How did it get there? So fiber is one of those things that it's not a vitamin, it's not a mineral. I consider it an essential nutrient, but I've been in lots of discussions where people can argue the other side. So what do we have for dietary fiber and how did it happen? So we have an adequate intake for dietary fiber, and it's based on epidemiological evidence that fiber protects against cardiovascular disease, which I'll show you in a second. So it's a different way of getting a number, a requirement, and adequate intake for dietary fiber. Then we have a daily value, and this is for labeling purposes. What's the daily value for fiber? It is 28 grams per day, based on 14 grams per 1,000 kcals, which I will also show you how that gets calculated in a minute. On the nutrition facts panel, this is US regulations. We have to have total dietary fiber, and we can have soluble and insoluble. And to get those numbers, we need analytical accepted methods. So as you see, as you go down this path, if we have a number and we can't measure it and put it on a label, we got a problem. We have to agree on those methods. Usually intake in the US is about 15, so it's about half. So it is a nutrient of concern, which makes it a high importance that people do not get enough dietary fiber. So I will show you this. This is really back in 2001 when the DRIs were put together for dietary fiber. And this is the relationship between intake of fiber in these prospective cohort studies and risk of developing cardiovascular disease. So you see relative risk on your y-axis and your energy adjusted dietary fiber on your x-axis. And a pretty interesting relationship. Lower fiber intakes are associated with increased risk, and higher fiber intakes are associated with protection or decreased risk. And so when you look at where do we come up with the 14 grams of fiber per 1,000 calories, it was based on this calculation that was done with modeling. And that is how we got to 28 grams as the recommended intake for dietary fiber. But if you are interested in live microbes and say, how would we get to an RDA or an adequate intake for live microbes? We have to go through the same process, basically. First of all, we have to prove we're an essential nutrient or food component, that we can't live without you. You are essential. And as you think about this as a dietitian nutritionist, that's kind of a heavy lift right there. Can we live without fermented foods, or do they just have some health benefits? How can we go down that path? Next thing we really need is some validated intake data that shows that intake of a nutrient or a food component is linked to a health outcome in healthy people, or disease risk reduction, which you saw for dietary fiber. And remember, this isn't like clinical data for sick people. This is in healthy people. Can we figure out how much of a nutrient do we need? Some of the things that can help, obviously, make our case that's important, and we need to fund research in this. A nutrient of concern. So dietary fiber is for American. As long as fiber is a nutrient of concern, that puts it in an important category. Also, do I want to get listed on the nutrition facts panel on a daily value? Obviously, most people would want that for live microbes. What is the daily value? Getting to that is another calculation. And then obviously, I'd love to get on myplate.gov, because that drives recommendations for government feeding programs and other recommendations. So is there any way I can get myself on the plate to prove that it needs to be part of the diet? All right, so let's go down the RDA path for live microbes. We need, first of all, an estimate of live microbe intake in representative populations or exposure. And you think, well, I know a lot of people, yogurt, kimchi, I know, you know, fermented foods, I see that, but we have to be able to measure that in a representative population. So Maria will talk more about how we figured out exposure, ways of getting at that answer. And then we actually have to figure out how to measure live microbes in food samples. So we can generate new data to put in databases so we can ask these questions with relationships. Obviously, we need a link to a health outcome or a disease risk in a healthy population with intake of live microbes. So I talked about in fiber, it is less cardiovascular disease. And a lot of fiber has many other effects, but that is the one that was used to come up with the adequate intake for dietary fiber. Strongest epidemiological studies are prospective cohort studies, which were the ones that were done on dietary fiber and risk of cardiovascular disease. So we would like to eventually get to that next study to build our base, build our support that live microbes are associated with an improvement in a health effect. And then continued scientific support that microbes are essential dietary components by authoritative bodies. So getting authoritative bodies to see this new information and move forward on recommendations. I use this and also from my book is that most everything comes into the body, right? So you're eating food, you just ate some lunch, and what happens is carbohydrates, proteins, fats get digested and absorbed, get into the human host. What goes down the other path and gets down to the large intestine? Mostly dietary fiber, right? So dietary fiber is going to get there, get fermented by the intestinal microbes, and lots of things are happening. So we're getting short chain fatty acids, vitamins get produced, and all of these have health benefits, but it's a pretty complicated system to show what is it beyond fiber that live microbes might have a role in this system. Just another review on how fiber affects everything throughout the gut. So obviously we focus on that fermentation, the short chain fatty acids, microbiota changes, pH down there in the large intestine, but throughout the digestive tract, fiber has health benefits and physiological effects. So chewing, slowing down gastric emptying, slowing down absorption of glucose and insulin, changes in gut hormones. All these things are potential mechanisms of why fiber has all these health benefits. It's a review that I was involved in, and just appreciating too that fiber has short-term benefits and long-term benefits. And as we're thinking about what are the health benefits or disease risks that we want to focus on, it's always difficult because obviously a lot of what we're interested in live microbes is immune. So some of these short-term benefits on the immune system we know for fiber. Interesting, certain fibers increase mineral absorption, and some of the fibers that have been accepted in FDA's new system, increased calcium absorption is the health benefit. When people are like, I thought fiber decreased minerals. Well, some of the older fiber studies show that certain fibers can decrease mineral absorption, but a lot of the oligosaccharide, fructooligosaccharides, actually galacto-oligosaccharides increase calcium absorption. Since calcium is a nutrient of concern, that's a health benefit that can be used for claims. And then just other autoimmune diseases, very hard to study. But also we talked about cardiovascular disease being the thing where we got our fiber recommendation. And obviously, why do we eat fiber? Because of its role in gut health, but that was not used in the support for getting the adequate intake for fiber. I've been involved in fiber for 40 years, and it's really fun now that people care. So, and I think, why did they get excited? And I usually go back to this paper from 2006 that showed that the gut microbes were associated with obesity. And if you read this study published in Nature, a very highly ranked journal, 12 unrelated obese subjects were fed controlled diets, fat or carbohydrate restriction. And when they looked at success in losing weight, it really did relate to an increase in bacteroides, correlating with a change in weight, which people said, hey, this is pretty exciting that not only did the gut microbes have some advantages for gut health, they have some advantages for obesity. And this has been known for a long time. So you'll hear more from Maria about all of the links with the gut microbes and health benefits. So these are all the things we do. We've talked about those: increased short chain fatty acids, producing vitamins in the colon, biotin and vitamin K, metabolizing carcinogens, enhancing calcium absorption we just talked about, making sure pathogens don't get in, and developing a mature immune system. All of these importance. And where do we get microbes? Obviously, we can get them directly from the foods we consume, or by eating fiber and other fermentable carbohydrates, we can make more and change the microbiota within the gut. So here's the question when we were asked this, and remember, this has been a long-term process, so this did not happen overnight. Is there a way to get for a recommended intake for live microbes? Because it's really of interest to consumers. How much do I need? What's a good dose? What do I expect from these different doses? Lots of questions about that. So as we went down, obviously it's not a vitamin, it's not a mineral, we don't have a deficiency disease, we have to think of it in a different way. So is there a framework for determining recommended intake of live microbes? And I want to really appreciate IFANS as a group that brings together experts to answer questions like this. So Johanna, one of our people, our leader today, but some important nutrition people got together with an ad hoc working group to come up with, how do we get there? You know, these are people that have been on the RDA committees and have been on the dietary guidelines to move the agenda forward. How do we get there? So they published a paper in Advances in Nutrition in 2021, looking at a framework for developing recommended intakes of bioactive dietary supplements. So, you know, as we've talked about here, vitamins, minerals, proteins, we can get an RDA. We got a adequate intake for fiber, but how about some of these bioactive dietary substances that don't fit into those normal categories we think about as essential nutrients? How do we go through setting up a framework for moving that agenda forward? So these were the 4 steps that they talked about to come up with intake recommendations. So first of all, we have to characterize the substance. So what is it? We have to be able to measure it. What is it? What are we looking at? And this is obviously the microbiota. Microbes are quite complicated, so you'll hear more from Maria on that. Safety is really important. We can't be recommending something that could be toxic. You know, we talked about the ULs and the whole process for dietary reference intakes. We want to make sure there's no safety aspects to these bioactives or other components that we would like to get an intake recommendation for. Is there some efficacy? What happens, right? What do we see? And obviously we talked about for fiber, it was lowering risk of cardiovascular disease. That shows that there's a health benefit or a decrease in disease risk. So we need that data. And then really just deciding, you know, the next thing, how do we set a number and how do we support that number? So, you know, it's a very long process to get there. And so when people immediately say, hey, we need a recommendation, we need consumer information, I hope you'll appreciate, as you hear the other 2 speakers today, absolutely, we do need to get there. And hopefully a lot of what we're going to talk about today will convince you that there are lots of things going on in that area. All right, so here's just an example of a recommendation, and we want moderate or high quality evidence to make that. So we need some good scientific evidence, and then we would recommend that you consume between, and then remember with units, CFUs, other units that we would use for live microbes of daily dietary bioactive. And is it foods? Is there anything, is it supplements? You know, both of those things, if we decide what our compound is, we would put it in there to support. What does it do? Is there a structure-function relationship, or does it reduce risk among the general population? So that would be an idea of where we would want to get with where we're going. So next steps, the good thing about this is brought together people that work, the nutritionists, food scientists, microbiologists, epidemiologists, food producers to develop a strategy to answer, can there be an RDA for live microbes, and what are the steps to get to that? We need to determine exposure to live microbes. You'll hear more about that. Estimating exposure, adding that data to nutrient databases. So if you want to ask a question, how much of live microbes are in the oranges, fermented oranges I just had, that's the kind of information we need to get into nutrient databases to even look at these relationships. And obviously in the US, we're lucky enough to have NHANES. So you'll hear more about some research that we've gotten done on that. So I will turn it back over to Marie.
No, you're going to turn it back to me.
Okay, Johanna, it's you.
I'm in charge now.
It's always good to have a boss. I always appreciate a boss.
Our second speaker is another wonderful speaker, and it's Maria Marco. Dr. Marco is professor in the Department of Food Science and Technology and chair of the Food Science graduate group at another wonderful university, University of California at Davis. She's had over 20 years of experience investigating probiotics, fermented foods, and the gut microbiome. And she's currently the vice president of the International Scientific Association for Probiotics and Prebiotics, and they have a good newsletter. That's a nonprofit organization that's dedicated to communicating the science of biotics. So Dr. Marco is going to be giving us an overview of the state of the evidence that live microbes can be beneficial to health. Welcome, Dr. Marco. Thanks for coming.
Thanks so much, Joanna, and it's really my pleasure and honor to be here with you and my fabulous colleagues in this question we're exploring of having live microbes, or at least more of them, in our diets. And so we had such a nice introduction from Joanna, and I'm going to be revisiting this topic of what are live microbes in the first place, and and give you some more recent studies that we've been doing to within ISAP and IFANS and to explore this topic. So here are my disclosures. I'm funded by federal international agencies and foundations, and as you've heard, I'm a volunteer in ISAP. And on the board of directors, and we're really, our purpose is to advance the science of biotics broadly, fermented foods, pro, pre, post, and so on. And as we already heard, I think we're at a really pivotal moment in time where mainly through technological advances, we have this new window into our microbial world. And microbes have been on Earth, well, much longer than we have, and in fact, we've evolved with them. And this is never more evident than if we look at our own body's microbiomes. And I love to share statistics. And so if we add up all the genetic contents of the microbes on our bodies, we're less than 1% human, a very humbling statistic. And just for bacteria alone, the number of bacterial cells on our bodies is about equal to our own cells. And again, there are more microbes on us right now than individually than there ever have been people on the planet. So another, I think, astounding kind of statistic that puts all of this in perspective. And if you look at the image here, you see that there's a lot of more density in the digestive tract of this individual. And that is indeed the case in reality, in that our large intestine is really home to trillions of microorganisms. And as already introduced, these microbes are really important for our health and contributing benefits from food digestion, vitamin synthesis, to having effects more systemically on our body through our immune system, and even affecting our mood and behaviors through our gut-brain axis. If you're following the microbiome field, you will also see that our gut microbes, or our body's microbes, aren't always associated or positive of good health, and so just some conditions and chronic acute and chronic diseases are shown here. So it's not always a good story, right? So and what we're trying to achieve in thinking about our microbiomes is moving our microbes more to the left of the screen and doing the good rather than harm. And as already explained, this idea of fiber reaching our large intestine and feeding our good gut microbes is a theme we hear again and again, and coming out in clinical trials and studies that show how fiber is not only really important for our overall health, but really good for our good microbes. And so that leads us to the question of, well, what else should we be eating that is really good for our digestive tract, for our systemic, for our health generally? And so, of course, the topic for today is microbes. And this isn't a new idea. It's been covered in New York Times years ago, in New England Journal of Medicine, that exposures to non-harmful microbes, such as in our environment, consumption of a probiotic, can be potentially beneficial for our immune systems and reduce the incidence of allergies. So what do I mean by safe live dietary microbes? Well, they'll come in different flavors, different foods. And so fermented foods, probiotics, and other food commensals. So, so that we're all on the same page, I'm going to take you through quickly these different categories. Fermented foods are foods and beverages made through intentioned or desired microbial growth and enzymatic conversion of food components. So to have a fermented food means there is microbial growth and metabolism involved in some step. And just to make sure, again, we're all on the same page, not all fermented foods will contain living microbes when we eat them, although they're needed to make the food. If you bake bread or some fermented foods are filtered or roasted, and for example, for coffee and chocolate beans. But those foods on the left, yogurt, kefir, cheese, kimchi, you can see will contain live microbes when we consume them. And another statistics, so we put, again, this in perspective, just as we have many microbes in our bodies, there are as many live bacteria in a cup of yogurt as there are people on the planet. Probiotics, this other category, these are different than fermented foods, the microbes in fermented foods. Probiotics are really strains at the very deepest genetic level. These are that are tested in clinical trials that have shown a health benefit. Okay, so the microbes we eat in a fermented food would not be in this category. And probiotics have long been studied. There's well over 1,000 human studies on all sorts of health endpoints. And some are shown here, and you can see if you go through this figure, you see many of the ways that probiotics have been shown to be useful or infecting our digestive tract health. But certainly there are other studies and the kind of emerging science of the use of probiotics for all other kinds of conditions well beyond the digestive tract. And mainly these are strains of lactobacilli and bifidobacteria. When you look at probiotics, these are the ones you're most likely to see. And so the third category, if we talk about live microbes in foods, is the microbes we ingest when we eat fresh produce. They're not as abundant as we would have in a fermented food with live microbes or in a probiotic, but if we have a salad, we are consuming a fair number of these microbes. And what you're looking at here in this colorful chart would be over 200 romaine lettuce samples that we looked at, collected here in California, and where we get a lot of the fresh produce. These are commensal non-harmful microbes that are just populating on our the fruits and vegetables that we eat. And so we're talking about thousands to millions of bacteria, and the way we express this is in colony forming units, or CFU, and so you'll see this maybe later when you look at this type of literature. So this brings us to the hypothesis that maybe consuming live dietary microbes in foods can benefit our health. And the initiative that that brought us here today was started by Colin Hill in a commentary published a few years ago, where he postulated that in addition to RDAs for nutrients and vitamins, dietary guidelines should also advise consumers to include safe microbes in their diets, such as with consumption of fermented foods. So this set off a whole series of events, but just to reflect on a moment why why we're here and thinking about this, and it's not an idea that started in 2018, but sort of reached maturation, I think, with the idea of our knowledge now on our own microbiomes. But long ago, there have been hypotheses of the old friends or biodiversity hypothesis that, you know, as we've become cleaner in sanitation and our foods and our environment, we are reducing our exposures to non-harmful microbes. So this decline in diversity can lead to microbial deprivation and putting our immune system out of whack. And this tracks with the rise over the past 50 years or so in immune-related disorders, asthma, eczema, for example, as well as the rise in gut-related diseases like inflammatory bowel disease. And to put this into a picture, we reflect back on our ancestors' diet that was not as sanitized as what we have now, clearly, with few ways to preserve foods, and I'm sure we're not having that super clean apple, you know, foods were different. We had exposures to microbes that could have trained the immune system or enhance systemic and gut health. And then today, our more modern diet with low microbial diversity and exposures. And then the idea that we're exploring is if we have micro-rich diet, we can retrain and our immune system that may benefit our overall health and well-being. So that leads us to the ISAP, International Scientific Association for Probiotic and Prebiotics work initiative that has again been going on for a few years. Collins and Hill's involved in this, and Joanne and myself and others you can see on the screen. And we just started by setting this hypothesis out to should there be the recommended daily intake for microbes and ideas how to test this to get an answer. So we started on the journey by estimating consumption of foods containing live microbes in the U.S. and correlating this consumption with disease risk. So next I'm going to go briefly over what we did and the results. So as Joanne mentioned, we used NHANES database. This is the National Health and Nutrition Examination Survey. It's a cross-sectional survey to assess the health and nutrition status of adults and children in the U.S. There's a large number of participants, and data are collected based on dietary data are collected based on a 24-hour recall, and this is every other year. And NHANES data allows us to correlate food consumption health status. So the very first question, actually, in using NHANES goes to, well, which foods contain microbes? That's not in the database. And so myself and several others who are experts in this field of food fermentations and probiotics, and mental microbes on plants, we went through the over 9,000 food codes and ranked the foods, whether we expected to have any microbes, medium numbers, high, and you can see the types of foods that would go into these different categories. And if you think about your own diet, it's probably no surprise that most, 96% of the foods would be in this low microbe category. So CFU, that coliforming units per gram, the approximate number of cells. The medium foods, so it's about 3%, and the high, fermented foods that, again, in the NHANES food or dietary recall database, 1% of the foods there would be in this high microbe category. So over 10,000 or 10 million microbes per gram. So what was the outcome of this? First populated this, and then the next question we asked, well, how many of us are eating these foods, and what is the daily intake in grams of these foods? And so the first finding, and looking over about 18 years worth of data, is that only about 2/3 of the US population eats any microbe-containing food on a daily basis. So only about 20% of consumers eat high microbe foods. So these would those in the fermentation, fermented foods category, whereas approximately 60% eat medium or high foods, according to what was reported. The second key point of the study is that we eat around 100 grams of microbe-containing foods per day for those who are consuming these foods. And sort of back of the envelope calculation, that comes to approximately 10 to the 9th microbes per day. And most of these would be from fermented foods. So up to, say, 1 billion microbes per day. And interestingly, if you look at the age range in the table, you see as you get older, you see more consumption of foods and a higher intake of these foods as we get older. So potentially, we have an opportunity here for younger individuals to increase this in their diets. And so that led us to the second question: is this related to health? And as explained, NHANES includes interviews, physical exams, and health assessment data that we could use to correlate or associate with live microbe food intake. So using regression analysis, we looked at these several parameters, not all of them here because many health indicators are measured, but more focused on cardiometabolic health. I'm not going to go into the data, and mostly because of a lack of time today, but I encourage you to interested to go to the paper. It's a rich data set where through regression found associations with these cardiometabolic factors and food intake, and with high numbers of microbes, medium, high, and fermented foods. So what you're looking at here are associations based on fermented food intake. And what hopefully is obvious is that there is a positive correlation between these anthropomorphic factors and biomarkers for cardiovascular disease risk, in the direction that increased intake of fermented foods results in reduction of risk. And that was true for all of these biomarkers except for weight, which was no significant difference. So some high-level outcomes of this study is that consumption of foods that contain high levels of live microbes is associated with a range of improved health benefits. And another message is that these benefits can be achieved by modest changes in diet, such as adding a yogurt into your diet or eating more fresh fruits and vegetables. We also, however, point out that this does not prove causality. And so that leads me to the challenges that we still need to address, and that is hopefully obvious to you, is distinguishing between live microbes and the food nutrients. These foods that we're looking at here are already regarded to be healthy for other reasons, and the fiber or the phytochemicals, the benefits that they confer. We're just adding microbes into the equation. And another challenge or aspect that we didn't have the opportunity to address is distinguishing between the types of microbes and the health benefits that they could confer. And ultimately, we would encourage the community for developing more randomized controlled trials and other forms of clinical evidence. So maybe we're not that far with that, despite this picture of sort of a conservative outlook based on where we stand today. But we could see, I think, not just from studying on probiotics and as a source of live microbes, but from the rich history of consuming fermented foods throughout the millennia, that they should or could be a part of our advised food intake on a daily basis. So I want to leave with the acknowledgements of our work group and ISAP and Marie and IFANS for the live microbes subgroup work group. And if you're interested in an overview or shareable infographic, this is one we put together on live dietary microbes in ISAP. And I will leave it there.
Maria, thank you. That was just great. Very, very interesting. And lots of questions and lots of good answers. Well, we're going on to our last speaker, and we're lucky to have our colleague from Canada, from Vancouver, Desiree Nielsen, who's going to wind up our speaking program and then get your questions into the question box so we know what your interests are. Desiree is a registered dietitian, and she's also the founder of Desiree Nielsen Nutrition, and that's a private practice and consulting business with over a decade of experience now in nutrition communications. And she's also the host of All Sorts podcasts. Her practice focuses largely on complex chronic digestive and inflammatory disease and also plant-based nutrition. So what Desiree is going to do is to provide some information on where the live microbes occur in the food supply, in the whole diet. And she'll also give us some practical guidelines on how each and every individual can benefit by incorporating these sources into the diet. Desiree?
So I'm really pleased to be here with you all today. I have been a dietitian for almost 15 years now, working in private practice. And, you know, for many of those 15 years, probiotics and fermented foods have been a hot topic. First, some disclosures. Like I mentioned, I have my own private practice and consulting business, and as part of that work, I have been a collaborator educator for many brands across the probiotic, fermented foods, as well as whole grain space. So what I find as a dietitian is that, you know, we've seen in the previous two presentations is that we have so much evidence to create a theoretical basis for why we want live microbes in our diet. The challenge is, as registered dietitians, we have patients and we have clients and consumers coming to us on a daily basis with questions about these, because there is so much talk about it in the media, on social media. And so it's really about closing the practice gap. How do we get from the theoretical basis, from the evidence we have in the science, to answering the questions that are being asked of us every single day by real people? So what is the awareness on the consumer side of fermented foods? What is the awareness on the health professional side? Do we have evidence of benefit? How do we guide? How do we tell people, well, here's how much you eat now, versus, you know, when we have better data 5 years from now, 10 years from now? And then for us as dietitians, do we have the knowledge required? Do we have knowledge of food preparation, of culturally appropriate and available fermented foods? And so one of the biggest challenges, and this happens over and over again, in the research versus what's happening in mainstream is that consumers are like the cat is out of the bag, and consumers are well aware of the potential benefits of fermented foods and probiotics. So there's a lot of talk of, take probiotics for X, take probiotics for Y. No, don't take probiotics, just eat fermented foods. And the vast majority of this awareness tends to be over a really small set of fermented foods, with yogurt absolutely remaining dominant in the North American market, with other foods having sort of like a smaller awareness. And one of the challenges with having just enough knowledge to be aware of these things is that consumers are very vulnerable to misinformation, very marketing-driven communications. If you ask people about probiotics, they can probably name one main brand that's being advertised to them by every single podcast and influencer on Instagram right now. And one of the challenges about it being such a large part of the common conversation is that in addition to having access to a lot of misinformation and believing things to be true which are not true about fermented foods and probiotics, the expectations of benefit greatly exceed the scientific evidence. So we'll have people saying, well, I drink kombucha every single day. Like, why am I still bloated? Or why is my IBS a problem? I thought I was getting my probiotics from kombucha. But health professionals are not immune to this either. I have been working with health professionals and educating on probiotics and the gut microbiome in fermented foods for a very long time now, with physicians, pharmacists, and dietitians. And we also vary widely in our knowledge of fermented foods. And a systematic review that was published just recently looked into the data of, pharmacists, nurses, and physicians on what is their knowledge of fermented foods and probiotics, what are their recommendations around these. And one study that was reported in this review mentioned that, the knowledge sources are not just primary research. Even as evidence-based health professionals, sometimes we're not just looking at the research. We are getting information from our colleagues. We are getting information from patients. And because we are also consumers of media, we are getting information from the media. And one of the studies reported in this review that physicians who recommended probiotics tended to refer more often to scientific sources, whereas those who did not rely on media sources more often. And so I just want to do a little quick review. Dr. Marco already talked about this. Because as I mentioned, this idea of, well, I got my probiotics because I consume kombucha every day or yogurt every day on a daily basis. And so there is a difference between fermented foods and probiotics. So fermented foods are the foods produced through that desired microbial growth, whereas probiotics are typically isolated live microorganisms, often in supplemental form. The microbial composition may vary. Hopefully in probiotics, we're getting more of a defined microbial composition, usually in a higher CFU count. So your dosage is going to be smaller volume versus fermented foods. And with fermented foods versus probiotics, as Dr. Marco mentioned, there's over 1,000 human studies done on probiotics to suggest outcome, or sometimes lack of outcome. But we do have less research with fermented foods with respect to concrete health outcomes. So why recommend microbes? If we are at this point in our knowledge base, why would we as health professionals recommend microbes? Because despite the many questions we still have, evidence is suggestive of benefit in several conditions, particularly for probiotics, because we have a far more robust human clinical trial cache than we do for fermented foods. So irritable bowel syndrome, prevention of antibiotic-associated diarrhea, prevention of Clostridioides difficile-associated diarrhea, as well as prevention of upper respiratory infection. And all of these are occurring at the systematic review and meta-analysis, and many of these have Cochrane reviews to guide our practice. And then again, with fermented foods, what's really interesting, particularly because we think of fermented foods as being for digestive health because of their potential impact on the gut microbiome and everything that's associated with that. The human evidence that we have so far in fermented foods is actually most robust, if we can call it that, with respect to cardiometabolic health. So then we get into the question, well, how much do we need to eat? How do we know, like, what is a serving size and what are the things that we need to think about in this with respect to this? And so, research that's already been presented, when we look at like how are we talking about servings or how are we quantifying fermented food intake, we had in the first, we were looking at low, so the typical sort of processed North American diet, medium, which is those fruits and vegetables, fresh, live foods, or any high microbe foods, which are the fermented foods that have not been heat treated or processed to kill the microbes within them. And then in the Hill trial, we were looking at 100 grams of fermented foods. But then we have this. And if you are a probiotic nerd and a fermented food nerd like I am, you've probably heard of this one. This RCT came out of the Sonnenberg lab at Stanford, and it was very well publicized. So if you are in these little niches in media and on social media, you heard about this trial, and many health professionals, myself included, love to talk about this one because it was really well designed. So we had, a very small study population. It was only 36. It was 17 weeks duration with a baseline intake, a ramp up to the study treatment, 6 weeks of intervention and 4 weeks of free choice afterwards. And what this trial did was look at high fiber versus high fermented foods groups. And you'll notice, earlier we mentioned that the average American is only consuming 15 grams of fiber a day. So our high fiber treatment group, their baseline was already higher than the average American. So they started out at baseline at around 20 grams of fiber a day, and the intervention got them up to about 45 grams of fiber a day. Whereas, and this comes right in line with the data that we've already been shown, for those in the high fermented food group, the baseline was 0.4 servings a day. North Americans are not consuming fermented foods very often, if at all. But through the intervention, they got up to 6.3 servings a day. And what was really interesting with the outcomes of this trial is that, for many of us, who myself included, who are huge proponents of dietary fiber, we know the evidence base on the potential health benefits of fermented foods and fiber. It was the fermented foods group that over the treatment time increased their diversity of gut microbes, as well as decreased almost every single inflammatory marker that was tested in this study. And this didn't actually occur in the high fiber group. And there was a bunch of reasons why this could have been. The first being that their baseline was already much higher than the average population, and that in such a short treatment time, it's possible that their body's ability to adapt, to truly harness what was going on with the excessive amount of fiber, because 45 grams of fiber a day is a lot, that perhaps they needed more time for their digestive tracts and their microbiomes to adjust in order to see benefit. But what I really wanted to pull out with this study was, here they actually define what is a serving and something different than just simply the presence of microbe-associated foods or in 100 grams. So the fermented foods were defined as following. So things like kombucha, yogurt, kefir, buttermilk, it was a 6-ounce serving, whereas the fermented vegetables like kimchi and sauerkraut, a 1/4 cup. And then vegetable brine drinks, so like the sauerkraut juice, which you can get as like a quote unquote gut shot on the market, was 2 ounces. And for us as dietitians, it's really important that we have clear guidelines as to what constitutes a serving of fermented foods. And the question is, how do we do this? What is our measure going to be? Are we going to quantify this by how many microbes we're consuming? Are we going to try and get to 1 billion a day or 10 billion a day versus what is a reasonable serving size of the food? And this is important because there is a wide variation in the microbe content of even similar foods between kimchis or kombuchas or sauerkrauts, for example. And then the other thing that we have to consider with a lot, particularly vegetable ferments, is that there's a heavy-handed dose of salt used in the fermentation. And so, this is a national brand of sauerkraut here, and a 1/4 cup of the sauerkraut contains over 500 milligrams of sodium, which considering, you know, the amount of people who are already consuming excessive amounts of sodium and the people at risk for cardiometabolic diseases, this is something that we can't just sort of like shove under the rug as we are like trying to get our microbe counts up. And from a nutritional perspective, I also really wanted to highlight, so what are the benefits? Yes, we have microbes. Yes, we understand that many of these fermented foods, the substrate is already very nutrient dense foods like fruits, like vegetables, like teas. But fermentation also changes the nutrition within foods through the fermentation process. So non-nutritive substances like fibers and resistant starches are broken down. They are more bioavailable after the fermentation process. Substances like phytic acid, which of course are going to bind minerals, that will be degraded within the fermentation process. By potentially enhancing mineral bioavailability, which for people on plant-based diets having more bioavailable iron, for example, can be a real nutritional benefit. Nutrients can also be transformed through the process. So proteins like casein and gluten are broken down. Fructans for those on a low FODMAP diet, which is why we see anyone who does low FODMAP counseling, traditionally fermented sourdough bread, wheat having a high fructan content, sourdough bread is deemed low FODMAP and acceptable on a low FODMAP elimination protocol. Production of GABA, things like GABA in fermented grain, production of B vitamins like folate can occur in the fermentation process. Increase in antioxidant phytochemicals like chlorogenic acid. Of course, as we have fermentation and the production of short chain fatty acids, the pH will decrease and enzymes can be produced. There are also other therapeutic considerations. So immunocompetence is very essential for the safe consumption of fermented foods, you know, and so anyone with significant autoimmunity, transplant recipients who are on immunosuppressive drugs, people undergoing oncological treatment as well, that is a potential risk factor for safe consumption of fermented foods. And not to mention, when people are fermenting at home, improper fermentation may increase risk of foodborne illness. And this is why for some, home fermentation may not, unless, you know, they've been doing this, you know, since their grandmother taught them how to ferment years ago. Home fermentation might not be right for some versus choosing a national brand with more sort of stringent controls. The other thing we have to think about is the intake of compounds with potentially detrimental effects. So I've already mentioned sodium, which is a hot button issue for us as dietitians. But in my world of digestive health and inflammatory conditions as we work within our practice, fermentation breeds the creation of biogenic amines like histamine, for example, that can cause issues for certain people with histamine intolerance. And then of course we have FODMAPs. So kombucha is not great for someone on a low FODMAP diet, but other fermentation products, as I mentioned, like a sourdough bread, which doesn't have live microbes, but it is produced through fermentation, can actually be beneficial on a low FODMAP diet. The other thing we have to think about is identifying culturally appropriate choices. So I wanted to share this framework from a group, a health authority here where I am from in Canada, in terms of cultural safety and also cultural humility. So it's very important for us as dietitians to understand our own personal and systemic biases so that we can develop and maintain respectful processes and relationships based on mutual trust. Understanding that we as dietitians always have more to learn, that our own culture and the frameworks within which we work create biases, and that we need to be curious and humble and open to learning from our patients and our clients, particularly with respect to the type of dietary recommendations we are making. Because while many of us know yogurt, kombucha, kimchi, sauerkraut, there are so many more fermented foods that are out there and culturally important to others. So it's really important for us, if we are going down this route of understanding that microbes may be of benefit, starting to ask our patients or our clients, so do you take probiotics? Do you consume fermented foods? Not simply giving a checklist of more Eurocentric or the fermented foods that we are aware of, but being curious. Are there fermented foods that you consumed growing up? What are the fermented foods that are culturally important to you? And having that awareness that fermented foods may not look like yogurt. in our clients or our patients' diet. And there are many more out there that will contribute to their intake of microbes. The other barrier is affordability and accessibility. So particularly for those of us in North America where kombucha, sauerkraut, kimchi are very trendy. They may be consumed and purchased at a health food store. These things can be very premium priced, and this may not be affordable, particularly considering how grocery prices are going up and grocery budgets are being squeezed. People might not have the available funds to add what is, essentially like a peripheral ring on their grocery bill when they're just struggling to afford fruits and vegetables and basic foods to feed themselves and their family. And this is where food skills can be really helpful, teaching the food skills necessary to ferment at home. Cabbage is a really cost-effective vegetable, and, with not a lot of knowledge, people can turn cabbage into sauerkraut at home for a fraction of the cost. So this is a way to get both more involved in food preparation, which is always a really important thing for all of us in terms of our health and well-being, but then allowing people to enjoy the benefits of fermented foods in a far more accessible and cost-effective way. We also have to assess appropriateness. So, what are the patient's nutritional and health goals? If they are concerned first and foremost with an acute condition and going through treatment for an acute condition, maybe we're not talking about microbes at all. This is not the time. But if someone is dealing with, if they're one of the roughly 11% of us who have irritable bowel syndrome, if they have something, that may be uniquely connected to a potential benefit from fermented foods and introducing live microbes into the system, is this in line with their health goals? Are they better served by focusing their efforts on increasing their intake of microbes or another facet of therapeutic nutrition? And remember that microbes are not just fermented foods. Absolutely, this is where we're going to see the highest concentration of microbes, but how's someone's fruit and veggie intake? This is also a viable and very real source of microbes vis-a-vis the, 96% of the foods in our food supply that are going to have a low microbe composition. Are the fermented foods already a part of their cultural foodways, and can we capitalize on something that's already culturally important to them versus bringing in a more Eurocentric approach and focusing on things like sauerkraut and yogurt? Are these foods accessible and affordable, and are there any contraindications? And with that, I will pass it off, and I think we are at questions now.
Thank you, Desiree. That was just wonderful. Just one question, and I don't think I'm the only one who wants the answer to this. What is kombucha?
So kombucha is a fermented tea, and I normally talk about different kinds of ferments. So we have ferments where we're simply harnessing the live microbes within the environment and on the food. We talked about, a sauerkraut, kimchi, we're not adding a defined microbe to that food. Kombucha is different. We use something called a SCOBY, which is a culture of both yeasts and bacteria that is added to tea, typically black tea with sugar to feed those microbes. And then we have both yeast and bacteria, so it creates an effervescent drink that's very rich in microbes.
Very good, thank you. Sounds good. Sounds almost as good as the wine in Napa, which is also fermented, I guess. Joanne and Maria, do you mind turning on your cameras as we go to the question and answer part of this? Oh, great. There's Joanne. Good. One other question to anybody. How do you know that all of these CPUs, these live microbes, are alive?
I'll take that one.
All righty.
And yeah. So when we talk about CFUs, these are, if you ever looked at a Petri dish, they're the little colonies. And to get a colony, you need to have one living cell. And that cell will divide and divide and divide, and then you get something you can see. And so when we talk about CFUs, we can say those are for sure alive. There is some nuance to this that confounds microbiology, is that we can't culture everything all the time. So we can be underrepresenting what's actually alive that's there in our CFU counts.
Okay, and how do I know it's in there when I buy the yogurt or the kombucha?
So I can take that one. So, you know, a lot of the times we don't, and that's a real issue. Most kombuchas don't list a CFU count on their label. There are, particularly with yogurt, we have labels that consumers can look for. So in the US, there's the voluntary live and active microbe symbol, which you might see on your probiotic yogurt. Here in Canada, when yogurt is labeled as probiotic, it's because there has been a defined Health Canada formulary probiotic microbe added in a specific amount to the yogurt itself. So for consumers, it is a bit of a challenge because most fermented foods aren't defining the microbes on their package. They're not defining the total amount of CFU count, but particularly around yogurt, where we've been doing this for a little bit longer, you can look for that live and active culture seal in the US, or in Canada, you can just look for the word probiotic because that has a Health Canada defined definition.
Thank you, Desiree. That's fascinating. Let's go to Joanne and put her on the hot spot on a couple of things. First of all, in the pursuit of recommended intake levels, how important is the population subgroup, like age, gender, race, and so forth, Joanne? And the other thing I noticed in the sort of the model for getting recommended intakes, you didn't have randomized clinical trials.
Obviously, the DRIs go for across the life cycle, right? So pregnancy, we would have to have information for all, but for starters, we would want to get it for an adult population, and that's kind of the NHANES signal. Can we get signal with NHANES data? So yeah, no, it's really challenging, and I think that one of the big issues we have is, and Desiree talked about this a lot, there are so many fermented foods out there across the world that how do we capture those? And then, you know, how do we say how many CFUs? Is there another way to give data? And we couldn't come up with them. As Maria, this has been a long, if I go back, the article she showed by Dr. Hill, he was the one that wrote the article and said, hey, let's get an RDA. And then unfortunately, I feel like I came in and kind of put a big blanket on the fire and said, hey, it's not going to be that easy. These are all the problems you're going to have just because of live microbes. You know, we just didn't have data. And I really appreciate the team that came together. You'll see Dr. Fulgoni, who's done a lot on NHANES data and coming up with how to even access that information. It's an incredible database to have these public databases that you can go in and access information and do these analysis and try to look at relationships, but there's no data there, right, for how much live microbes. So the amount of time, and I really, if you look at the people that did the, Maria talked about low, medium, and high, we didn't start there. You know, we really were hoping to have numbers for absolutely every food that we could come with in a nutrient data bank, and we couldn't get there. So to me, and how little consumption there is the other issue we have. Besides yogurt, which we have pretty good signal, people consume yogurt. As Desiree said too, there's all these other foods out there, but they're just in these large data sets, which is kind of building our base on the epi side. We just don't have very many people consuming. And a lot of people that are going into fermented foods are healthy people that are not maybe the population we want. So besides the different age group, we want to make sure that across populations, people that are not already committed to healthy diets, eating lots of fruits and vegetables, we can convince them that it's important to consume live microbes too.
Terrific. Thanks, Joanne. Maria, let's go on with that whole business about the healthy and unhealthy. There's a statement that autoimmune disease, I think it's the Autoimmune Disease Association put out, about reduced microbes. Reduced beneficial microbes should be explored. Apparently, some people say since it's known that this is a statement that somebody made, since it's known that beneficial microbes in certain genera are known to produce indoles that interact with the aryl hydrocarbon receptor, which affects immunity greatly. what are your thoughts on that comment? One of the listeners.
Mhm. Yeah, no, I appreciate the question, the comment about autoimmune disease and what we're finding regarding certain microbes and the production, the transformation of tryptophan and into indoles and how that signals through the body. So I really think we're at the nascent stages of this research, but it is really exciting and shows yet another example of how microbes in our intestine and microbes in our foods and transformations of our foods through fermentation can have these global effects that we're just starting to understand. I think it's an exciting field of research that we should be following.
Amen. And that Stanford study that was mentioned, those markers were really not of immunity, were they? They were of CRP and other inflammatory markers. So the problem is the inflammation issue and then the immunity.
And this leap that they've already made is, oh, this is cute knowledge if I pay for this and I have my microbiome sequenced. But we get into a whole bunch of things. The first is that the stool microbiome is not indicative of the microbiome of the gut in total. It's not the same thing. The other thing is that these companies now realizing, well, people might not feel there's a need to pay for, well, here is what your microbiome looks like. I don't know what to do with this. They're already jumping to making very specific dietary recommendations based on your microbiome right now. We don't actually have the data to say, if your microbiome is this, then you should consume microbe X, microbe Y in these amounts, or you shouldn't consume this food in this amount based on your current microbe situation. And so this is really important because our patients and our clients are curious about these testings. They are making behavioral changes based on the recommendations that these companies making money off the testing are providing, but we don't actually have robust data to say if your microbiome is X, then you do Y. And so the challenge is for us as health professionals, again, it's this huge practice gap. It's this chasm between the scientific evidence and this person sitting in front of us asking us for our guidance. So what we do know is that if people have issues such as Crohn's disease, ulcerative colitis, irritable bowel syndrome, where we know in the evidence there are changes to the microbiome that are likely contributing to their symptom profile, then we can make very boring, very standard nutrient recommendations that our clients are probably still not doing, which is how do we get their fiber intake up? How do we, if we need to modify the microbiome, we have the low FODMAP diet as a microbiome modifier in our therapeutic toolkit as well. And then yes, fermented foods and probiotics. We do have probiotics on the market, and a great resource for health practitioners, if you didn't know about it already, is the probiotic chart. So in Canada, it's probioticchart.com. In the US, it's usprobioticguide.org, I think, or .com. And what this is a third party. It's run by a clinical pharmacist out of Canada with a scientific advisory board of physicians and researchers that review the probiotics on the market. They delineate which microbes they contain, what the CFU count is, what the indication is, and the level of evidence. And so as health professionals, we are looking for products that have at least level 1 evidence, so at least one good randomized controlled trial in humans to justify their use. So we do have some tools at our disposal. However, because our microbiomes are so diverse, because we don't have the data to guide us in customizing and personalizing based on our microbiomes, the best we can do is say, here, this has an indication for IBS. Let's try it for 12 weeks. Let's assess benefit. Maybe there's no benefit. Maybe it doesn't work for you. And then we move on.
Terrific. Thank you. I think we're up to time. So I think I better turn it over to Marie for a final word. And if we have any extra time, we'll come back and get the final word from our speakers. Marie?
All right, thanks so much, Johanna. And a huge thank you to our speakers, as well as those of you in the audience who provided some great questions. And of course, we have more than we could manage. So for those of you who are seeking the CEU certificate, we'll provide access via email shortly after this webinar closes. And we encourage you to take a look at our website, including all of our funded research and the work of the Gut Health Committee that provided you this webinar today. And we look forward to seeing you at some of our future events. So we hope everyone has a great rest of your day.
Thank you so much, Marie, and thank you to all of the speakers. It was wonderful. Really enjoyed talking with you.